Hi, I'm Wendy Chung. I'm going to tell you this morning about some exciting advances we have in gene therapy. So, your child or your family member's condition is genetic, and the obvious question becomes, can we do something to fix that? Can we go in and edit the gene? Can we fix the gene? Can we add back a gene? And many of you have been asking that question. I'm going to give you an update today in terms of where we are. I certainly am optimistic that in the future there are going to be many options in terms of genetically and molecularly based therapy. But really, the $1,000,000 question is when. And I don't have a great answer to that, but I will say that there are many people working on this. And I'm going to try and demystify this a bit for you today by explaining some of the strategies. So on the left here is what we're showing as a cartoon of a normal gene. That gene encodes the instructions to make a protein. And again, on the left here is someone where that gene is working fine. Now, on the right, there's a gene that has a genetic change. It's shown as a broken gene here. And in some cases, it makes no protein. That gene just doesn't have instructions that work, so it makes no protein. In other cases, it may make a protein, but it looks different than the normal protein. And in some cases, this can lead to an individual with a genetic condition. And that's what we're going to be talking about today. So very broadly, depending on whether or not the problem with the gene is not making a protein or making an aberrant protein or a protein that looks different, there may be slightly different strategies. I'm going to go through the most common strategies that I think might work for your family member's gene, but to say that there are still evolving strategies. And so I won't have time to go over in all of the strategies or in great detail, but I'll go over the most common ones. So let's dig down for a second. So here on the left, shown in red, are individuals that have a gene that has this particular change in it, a change that may, in this particular case, result in disease, and it's because this protein is not made. In those cases, there can be different strategies, but strategies in which we add back a normal copy or a functional copy of the gene. This then makes protein that was missing or potentially not there in enough quantity, and then corrects the condition because we have added back what was missing. Now there's some subtleties in this. As we add back this functional copy of the gene, where does it go? Does it go directly within the genome or the genetic information of the person? Does it integrate or does it become part of them? Or is it something that's living off to the side in some way? And there are some important differences between that that have to do with whether or not we get it in the right place and whether or not it stays around for a long period of time. So those are some subtleties, but this is the basic idea. Now, in certain cases, I showed you in that picture, there may be a gene with a change depicted here. And in this case, it actually does make the protein, but in this case, the protein is not doing what we need it to do and may in fact disrupt or interfere or gum up the works of the regular gene. So it's doing something that we don't want it to be there doing. And in that case, we actually have to get rid of it. It's not simply replacing it or adding back, but it is actively getting rid of this red protein here. And so there are strategies. I'm going to use the term gene silencing, but it's one way or another of being able to knock down or get rid, and if we can, of doing it specifically for the form of the gene that's making the protein that we don't want to have there. And so in that case, the reason why we get correction is we are getting rid of this particular protein. Now, for doing that, there are different ways of being able to deliver what look like these little, I don't know, these little green ladders here, or half of a ladder. There's a way of being able to deliver it to the cells in various ways. I'm not going to focus on those details today, but it is critically important to know the difference between the case I described over here, where the gene, or the protein rather, is missing, and the case over here where the protein is present and we don't want that version of the protein there. Fundamentally very important to differentiate. And I'm glad to say that in the cases that we're going to be talking about for your gene, I think we're to the point of understanding those differences. So that's the good thing. Now, some of you have probably jumped to the obvious conclusion to say, Well, why don't, if the gene with the change is here, just fix it? Like surgeons, just go in and snip it out, take out the version that's not working, sew in the version that does work, edit it, correct it, make it whole again. And if we fundamentally correct the DNA itself, then it will correct the protein. We don't have to do anything fancier than that. We can just go in and get to the root cause or get to the heart of the problem. And so this is a technology people are using called gene editing. You may have heard the terms CRISPR, Cas9. Those are effectively like the molecular scissors to be able to go in and to find just the right place, just the right location to do the snipping and then to do the sewing in. This is certainly a technology that is evolving very rapidly. Some of you have heard about the improvement recently called prime editing, which increases the fidelity or the likelihood, excuse me, that the editing gets done in just the right place. Now, the opposite of that would be some editing that's promiscuous, where you don't get to just the right place, but you may get to some place down the road that you didn't mean to be editing, or someplace down the road over here that you didn't mean to be editing. And there is the possibility of misediting, actually putting in something or cutting and pasting in the wrong place, and that could actually be bad. That could actually disrupt a different gene that you weren't intending to touch and cause its own independent problems if it's not very precise, very accurate. So within this, as I said, there are more and more tools that are becoming available. Some of these use what we call vectors or ways of being able to deliver these tools to cells. I'm going to be talking about that in just a second. Some of them are delivering these tools, these scissors, the thread to be able to sew this in. And really, the good news is that as these tools are developed for one condition, they can be readily applied oftentimes to other conditions. So you may be hearing about methods that have to do with gene therapy in the eye, or gene therapy for sickle cell disease, or gene therapy for a liver disease, or a bleeding condition. The good thing is that we are going to shamelessly beg, borrow, we won't steal, but we will borrow from others in terms of as they develop those tools, we will use those same tools that we're thinking about for your family's condition as well. So we do have to be patient. And what I want to give you is a little bit of historical perspective. You don't need to read all of the details here, but simply to show the timeline. So when we first started thinking really in the modern era about gene therapy was about the time I started my career in genetics, about 30 years ago in 1989. Now, the good news is we've come a long way in the last 30 years. We've made major advances in being able to understand this, but we also have learned some very valuable lessons. And I'll just point out one valuable lesson we learned in 1999 was that this is something that could actually do harm, that we need to be careful about this because a young man by the name of Jesse Gelsinger was actually unfortunately passed away during his gene therapy for a different condition. From that, we became, as a community, internationally cautious. Excuse me, in terms of being able to be careful, be safe, have checks and balances, test things in cells, test things in other animals, but really to make sure that we were going to do no harm, that we were going to protect the safety of the individuals who went through gene therapy studies. And because of that, we've slowed down. We're not running quite as fast, but on the other hand, I can assure you that we're always have the participants' best interest in heart as we're taking those precautions. So let me tell you a little bit about how we get those genes into the cells. And there are some limitations based on these delivery mechanisms or these delivery systems that we have. So the first is that there is a virus called adeno-associated virus. And I'm showing you here this viral particle. Within this is something that is a virus that is part of what causes the common cold. We have done things to these, genetically engineered them so that you can't pass them from one person to another. So it's not like the colds, it's not like SARS-CoV-2. This is not something that gets infected from one person to another. We have basically paralyzed them so that can't happen. But they are good delivery systems that inside this little squiggly here is the gene that we're trying to insert. And so as we're doing this, as I said, we have inactivated these viruses. Importantly, this is a cell that I'm showing here, and this is the nucleus inside the shell that houses the genetic information. And these are meant to be chromosomes housing the genetic information. When this virus goes in and infects the cell, this genetic information for the new gene comes into the nucleus, but importantly, it does not integrate within the person's genetic information. So we call these non-integrating. By setting them up so that they are not integrating, they will not disrupt other genes that the person has. And one of the types of genes that we worry about in terms of disrupting are genes that are mission critical to the way your brain works or your heart works, and also genes that might protect you from cancer. We don't want this to result in changing one condition or trading one condition for another. We also have engineered these cells or used particular adeno-associated viruses that they hone in to particular parts of the body. And in fact, it's been quite successful, remarkably successful, at being able to hone into brain cells or to neurons. And that's been a huge advance. This is also something that works quite well for cells like brain cells, like neurons that are no longer dividing. Otherwise, if they were dividing, you can imagine this DNA that gets in would be diluted out over time. That is, that when this cell divided, you'd still only have one copy of that DNA, and therefore only half of the cells that would retain that DNA and make the protein. So because brain cells don't divide, this is a very good strategy. It stays there. As we have gone through these gene therapy trials, and this is true across many different conditions, part of the reason why it takes so long is because we are, as I said, trying to make sure that safety is paramount. It's a very, very important thing to us. And as we do this, we try and make sure that we get just the right dose of the virus. We don't want to give too much to the virus. We learned our lesson. We gave too much. Collectively, we as a community gave Jesse Gelsinger, the young man who died, he got too much of that virus. So we're very careful about that. We're careful about testing, as I said, in cells and what we call preclinical testing in animal models. We test to make sure that the gene gets there at high enough dosages of the genes, and then ultimately not just that it's safe, but that it actually works. It's efficacious. It has the desired beneficial effect to slow down the disease or even to be able to reverse the disease. So a lot of thought goes into this to doing it well. Now the success, the good news is, and I'm just going to cut down to this bottom two lines, there are already two medications approved, two gene therapy medications approved for two different conditions, one an eye disease or a retinal disease, another for a condition called spinal muscular atrophy or SMA. And I'm very happy to say I've personally been part of these SMA studies, and we can now treat babies, newborn babies in some cases, with the gene therapy. It's literally an intravenous infusion through a vein. We don't even have to do a direct injection into the brain. We can just give it into the vein as a one-time treatment. I hope that this is going to produce a durable effect, that this is going to be something that's going to last for decades and hopefully a lifetime. We're still only a few years into this, but it does already show to be quite promising. Although I'm not going to go into it today, there are many, many other conditions that are currently being studied in what we call clinical trials. In other words, we're still testing them. Other brain conditions, so importantly here, adrenoleukodystrophy, another brain condition that I think is going to give us significant experience in terms of how we can do this for neurological diseases. So coming to wrapping all of this up, there are many things that we consider, and we try and matchmake. We try and find for any one disease, understanding the mechanism of how does that protein have its effect based on the way that that protein is working or not working. Think about exactly what type of technology would be useful to be able to get the correct version of the gene in, be able to permanently keep it within the brain cells, and to make sure that it's safe. Now, as we're doing this, there are a few other additional things that we consider. We want to be able to get in just the right amount. So it's kind of like Goldilocks and the Three Bears. We don't want it to be too much. Don't want to be too little. We want to be just right. And that just right, we hope, is what's going to be both safe and efficacious. And that's still part of what we're working on as we're getting into brain cells, making sure that we're getting just the right amount, making sure that we're getting in the brain, we're uniformly getting it into all the cells across the brain. There may be some consequences if it's a patchwork, if it doesn't look like we get into all the brain cells uniformly, we're not sure what that impact is going to be. And so we do need to be thoughtful in terms of how to do this, as I said, evenly, make sure it lasts, make sure there are no adverse consequences or no complications. So as we're doing this, just to let you know, there have been folks that have been at this for decades. The good news is that I can feel a real acceleration in terms of the breakthroughs that we're having, being able to learn from other diseases, other neurological conditions, and importantly, the safety that we now see in terms of the types of vectors we're using, the viruses we're using to deliver this, and importantly, that this is something that looks like it is lasting so far. I'm getting, for all of us, excited that we need to get ready. We need to prepare so that when some of these breakthrough technologies are able to scale and be applied to other conditions, I want our communities to be front and center. I want us to be at the starting line, ready to go, ready to take off on this race. And so many of the things you're hearing about at this family meeting are how we as a community can prepare for that day. I don't know whether that day is going to be in 2021 or 2025. It's hard for me to say, but I do think it's going to be here much sooner than we might think, because we have made so much progress over the past 3 decades. So I'll stop here, but enjoy looking forward to the discussion.